Right. Good morning. My name is Ralph Lampman. I work for Yakama Nation Fisheries for the Pacific Lamprey project. Today I'll be talking about the uh, assessment of invasive species predation on Pacific Lamprey and Salmonid species using qPCR and metabar coding. We have lots of partners that are collaborating on this project, so you can see the entities here on this first slide. Just a quick outline, I'll be sharing the results from the initial hatchery trial in 2023. We also did a field study in 2023, so I'll be sharing the methods and results. Then we have some partial results from the 2024 study, so I'll be sharing that at the end. We are interested in lamprey population in the lower Columbia Basin. There's lots of things that impact lamprey. Predation is definitely an emerging one that seems to be more and more critical and important. So we are really interested to know how these invasive species in the Columbia River, mainly we studied the lower Columbia, a little bit in the Snake and Yakama River. Um, you know, we're interested in the, where these predation occur, when they occur, and what species are really the key ones. So we did a hatchery experiment at our Prosser hatchery, where we have lamprey that we hold. We focus on northern pike minnow and smallmouth bass. This is showing the qPCR result on the left side, metabar coding result on the right side for both northern pike minnow and smallmouth bass. The upper graphs are the foregut and the lower graphs are hindgut. So it's just showing the hours after digestion. So you can see that the detection concentration decreases over time. It was more evident with the qPCR than metabar coding, but overall there's kind of general decline of that detection, and then around 50 hours is when it seems to disappear. So when you combine the foregut and the hindgut, this is what the graph looks like. So it starts out high, around so in this eDNA detection is kind of an inverse relationship. So when it's around 40, that's when you have no detection. And when it gets close to zero, it's high. But you can see it declines to basically the absent value around 50 hours. And that's a lot longer than when what we could detect from a visual identification. Usually 0 to 6 hours, we can see the lamprey. This is a picture showing a 7 hours after it was digested. So you can see part of the skin's digested. The notochord is starting to show up, that little white line there. Some of the skin's still present. But after 6 to 12 hours, it does become pretty hard to identify. You would have to kind of look for like teeth and other features if it's a juvenile lamprey. Larval lamprey don't have teeth. So yeah, we can expand the detection duration by about eight times compared to the visual identification. This is showing a kind of a snapshot of what the lamprey looks like in the gut of northern pike minnow, smallmouth bass, walleye over time. So by the time it reaches 50 hours, it's just a little bit of tiny bit of liquid, small smear left, and anything in between through those different digestion hours. For methods, this is a molecular analysis and it requires a strict protocol and making sure we don't contaminate the samples. So we have relied on other studies that are collecting fish. So electrofishing or hook and line, juvenile traps, we try to use whatever was available. We insert ethanol initially into their mouth they're after they euthanize. This stops further digestion and helps preserve the samples. Predation, and then um, they're killed by physical injury to the head. Sorry, I think I messed up the order. So first we kill them through the physical injury, and then ethanol is inserted. We don't want them to be flopping around while we're trying to insert the ethanol. Then we freeze them, and we can do the dissection later, or we can go right into dissection depending on what's available. And then we cut open the digestive tract from esophagus all the way down to the end of the gut. And we could freeze it at this point, or we can also further, remove the remnants from the digestive tract and place it into a buffer solution at that time. So some pictures of what this looks like. The upper left is ethanol injection, and then the dissection. We have different sets of scissors and equipment for different parts of the gut, and some examples of what the gut looks like for the different species on the right. The pike minnow doesn't have a stomach, so it's just a long gut. And then I think for the case of walleye, we were not able to kill some of the samples, so we did a non-lethal gastric lavage and used funnels and mesh screens to help collect the remnants without killing fish. These are the sample sites for 2023. Our target species was smallmouth bass, northern pike minnow, walleye, largemouth bass, channel catfish, yellow perch, and just a little bit of American shad. These are the results of what was detected based on the sites. Pacific Lamprey is orange, so kind of highlighted some of the ones here. So we did have detection in both the Yakama and the lower Columbia. And the red is Chinook salmon, coho is green, so you can see those colors show up in there. Just by looking, at by predator species, what they're eating. This is kind of the overview. We only had like one or two samples for the shad and the yellow perch, so not kind of focusing on the 5 species in the middle. Pacific lamprey is green, light green, and the dark green is lampetra, so western river lamprey or western brook lamprey. You can see that the ones in the middle, which are the northern pike minnow, smallmouth bass, and walleye, had Pacific lamprey detected, and yeah, all 5 species had Chinook and coho, and some had a rainbow trout / steelhead detected as well. When you look at temporal trend, so we did the sampling in April, May, and June. You can see that the proportion of detection increased as it got warmer into May and June, except for maybe coho salmon. It was about the same in May and June, but everything else tripled or doubled, tripled, or quadrupled as it got warmer in June. We also used the results from the lab study where we were able to, create an equation to estimate digestive time. So with that, we analyze, what time lamprey might be getting eaten. And this is the results on the left showing the time is mostly like in the early mornings or kind of early to late evening. Uh, that seems to be kind of the key time when they're getting fed. Um, and and just by looking at different predator species, we can kind of break it down even further on the right. So northern pike minnow, smallmouth bass, walleye. Like most of the smallmouth bass feeding was in the early evening, or we predicted to be early evening, based on our model. And then walleye was feeding pretty much throughout the day, but yeah, a lot in the evening and early mornings. So kind of, yeah, 3 to 7 a.m. and 4 to 10 p.m. zone, except for that one outlier in the middle there. Um, and then when looking at visual ID versus metabar coding, um, you know, there's a lot more species that we're able to detect. So uh, you can see, all this, and metabar coding was focused on fish species. So that's what we use for this study. So these are all, fish species that we detected. Visual ID, it includes invertebrates and other things as well. So like shrimp um, and what else do we have? Like clams and invertebrates, eggs are there as well. But 72% were considered empty, but with metabar coding, only 35% were considered empty. And lamprey and detection increased from 1 to 6% with the use of the metabar coding. So now 2024, we are, in addition to the fish collection, we have started collecting avian samples and also sea land samples through the help of collaborators that are listed here. So I will share a little bit about those results. These are the sample sizes, so for kind of by months on the top headers, and then by species on the for rows there, so you can see um, a total of let's see here, about 913 samples. Majority were still fish, but also a lot of bird samples and bird excreta samples, basically bird poop and sea land samples. So this year we are interested in a few additional questions. You know, there are some bird or sorry, we with the ethanol injection, that has been our protocol, but there are other studies that don't use ethanol. They just freeze it right away or get it to cold temperature right away and preserve it. So we are looking at studying that further to see if we can just omit the ethanol injection and still get good results, comparable results. We are also interested in fecal samples, especially for birds. Some of the birds, like white pelicans, we can't um, take lethally, so we need to, use other methods to analyze those species. So we have started that, and and then also with the sea lion, we are uh, we were able to we were given gastric fluid sample from the gut. So it doesn't include big tissues or anything like that, but we're trying to see how this is compares with the other diet analysis that they do on the sea lion. So we have some results for those. Uh, these are some action shots of the avian samples, skulls, and the excreta sample, and a walleye sampling there. These are the locations. They're pretty similar to last year with some additional sites that were added, um, focusing on Lower Columbia and Yakama River, a little bit in the Snake River. As far as locations, these are kind of the major sites, kind of by dam or location, um, and then also by species. They're color coded. So um, avian fecal species are red, avian stomach is green, and fish stomach is light blue. Gastric fluid from sea lands are purple. A lot of the, yeah. And then just by months, these we started in April, but instead of ending in June, we went a little further this year. So July, August, a little bit in August, October, November. Most of the fall samples were the excreta samples from birds. Um, but yeah, just a little bit of extended duration. These are, we only analyze about just a little less than 200 samples so far. These are the detections that we got so far from all the different predators kind of combined. You can see, Chinook salmon's kind of the number one, coho salmon, rainbow trout, Pacific lamprey are all pretty similar. A little bit of white sturgeon, quite a bit of sculpin, um, mouth chub, sucker, and so forth there. Just to highlight, which species are native, kind of highlighted those in red. So about uh, 3/4 to 2/3 are native, and the rest are not native. And now am I doing okay with time?
Yeah, I got about 4 minutes left.
Okay, great. Just a few more slides. This is the results for the bird species, seagulls and white pelican. You can see that seagulls are eating more on the native species, but I just need to clarify this white pelican samples were mostly in the fall when, predator or salmonids and lamprey were not migrating. So definitely need to do this study, look at the results from this upcoming study to look at answer that question about native versus non-native. Sea lions, California lions versus stellar sea lion. You can see they're feeding mostly on native species, very little of the invasive species, non-native species. And smallmouth bass, walleye, northern pike minnow, walleye and smallmouth bass tend to feed more on the native species, and northern pike minnow is feeding more on the walleye, smallmouth bass so far with the limited samples that we have. But we'll take a look at the full samples when we get the funding, which should be coming very soon. So conclusions, the collaboration really enabled us to collect many samples for this study. The genetic techniques does show that they provide high sensitivity and longer detection window for lamprey predation, and genetic um, and let's see, yeah, and there's a temporal trend with April, May, June, kind of increasing trend over time, and most predators, invasive species, or non-native species were shown to feed on lamprey and salmonids. I think I covered most of these, so maybe we'll just move on to questions.